Welcome to the CHO Cell Line presentation. My name is Rafaela Diaz. I am a 6th semester student of Biotechnology Advanced at Seneca College, and today I'm gonna to show you that the immortality maybe is in your gonads. Chinese hamsters had been used in research for more than 100 years. In 1919, scientists began to use it instead of using mice in lab. And almost 50 years later, ovarian cells came up as an option to be cultured. CHO cells, which stands for Chinese hamster ovarian cells, were first established by Theodore Puck at Eleanor Roosevelt Institute at the University of Denver, Colorado in 1957. Since that time, several changes had happened. CHO cells are important due to the rapid growth in suspension, high protein production, and seem to ignore the half-life limit, as there is no limitation on doubling time. As an example, Puck grew those cells for more than 10 months with no decrease in the cell rate division and almost no change in the morphology. CHO cells are also prone to genetic modification, cell transfection, recombinant protein expression, and clone selection. For pharmaceutical applications, they are also important due to the ability to undergo post-translational modifications. Also, due to the low number of chromosomes, only 22, they are excellent for radiation cytogenetics and tissue culture. As well, they do not express the epidermal growth receptor factor, EGFR, which makes those cells great to investigate the mutations on those genes. Also, it lacks in proline, which will directly affect the production of p53 gene, which needs the proline to act against tumors. There are many CHO cells around the world. Today, we are going to focus on CHO-K1 and CHO-DG44. CHO-K1 was the first variation of CHO cell. It was originated from a clonal selection process and do not express epidermal growth receptor factor, making this strain a good option to investigate mutations in this factor. This strain called CHO-DG44 is widely used in industry due to the absence of the DHFR gene. As those cells do not have these genes, they can only grow if the media is supplemented with glycine, hypoxanthine, and thymidine. By knowing that, scientists can transfect the interesting gene with the DHFR fragment into CHO-DH44 cell. They then can take those recently transfected cells and try to grow in a media lacking on those three components. If the cells grow, it's a sign that the transfection worked. This seems to be a simple idea, but it makes the genetic manipulation very easy. If you check the specifications for CHO cells on ATCC, you'll be able to see the all variety of cells that are available over there. However, as today we are focusing on CHO-K1 and CHO-DG44, I'm going to talk specifically about it. CHO cells, in general, are epithelial adherent cells that show a doubling time of about 20 hours. Due to its characteristics, they are great for EGFR studies and tests involving the presence of the DHFR gene. As Burton et al. Observed in his study in 2010, CHO cells are susceptible to several viruses. However, when compared to the other types of cells, such as BHK cells, its susceptibility is considerably lower. To culture CHO cells is necessary an F12 media added with 10% of FBS, and depending on the strain being used, it is necessary to also supplement the media with some specific amino acids. As for example, the CHO-DG44 needs to be supplemented with 3 different amino acids. Cells need to be incubated at 37 degrees with an increment of 5 to 10% of CO2. This incubation has to be
in shake flask, spinner bottles, or tea flasks. When subculturing CHO cells, the split ratio generally goes to 1:5 or 1:10 when cultivating in flasks and 1:2.7 when cultivated in 6 well plates. When necessary to cryopreserve, it can be done using the vapor phase of liquid nitrogen using 1.5 to 2 ml vials. To sum up, CHO cells are a great option in industry due to the limitation on doubling time, high protein production, being able to transfect genes, and all its variations that allows many genetic manipulations. Thanks for watching!